once again, I'm called Rose Nakasi, a PhD student in computer science, Makerere University. I'm part of the team at the Artificial Intelligence and Data Science Laboratory. And this project comes through the Ocular team. Ocular, the reason why it was given that name, we want to leverage on the advantages of using a microscope for diagnosis. And right up there, one of the key components of the microscope is the ocular eyepiece or the ocular lens. So we want to improve how diagnosis is done through use of the microscope, but specifically improving how we view images through the eyepiece. What problem are we trying to solve at the AI lab through Ocular? One of the big health burdens in diagnostics are the very many numbers which usually get malaria. Malaria, first of all, is one of the killer diseases in the country. According to the health bulletin that was released in around 2016, it revealed that 27% of the lives of Ugandans we have deaths of Ugandans accounting to around 27%. So that is a very big burden. Microscopy, the work that we have done, though my work is specifically in malaria, we have scaled it up to tuberculosis, and we've also scaled it up to intestinal parasites. And we all know that tuberculosis is also one of the big infectious diseases we have in the country. Apart from that, we also realize that when diagnosing malaria, the gold standard method for diagnosis is the use of the microscope, because that is where the confirmatory test usually comes from. So it is a bigger component when we come up to improve it and make it a bit better. So right there in the pictorial, you see we have very many patients who flock the healthy facilities waiting for a diagnosis to be carried out. Usually in the presence of a standard diagnosis, when you get to the rural areas, you find that most of the people are diagnosed by just looking at the symptoms. Someone goes to a healthy facility because you have a headache, you diagnosed and you're told that you have malaria. So very many people go undiagnosed. Others, there are cases of misdiagnosis. So it is very important to keep the gold standard at a very high priority in the country. But there is a challenge using microscopy. We find that there was a study that was carried out in Ghana, which is one of the developing countries in Africa. We found that there's only 1.72 microscopes per 100 population. But then we only have around 0.85 trained laboratory staff per that 100,000 population to carry out the diagnosis. And what does that mean? That means that we have more microscopes in the developing world than the trained technicians to do the diagnosis. So right there you see a lab technician at Mulago, and behind there are the people who are waiting for a microscopy diagnosis. That means he's overloaded with a lot of patients that come in a day for diagnosis. And when you go back to the standard procedure for diagnosis, it only requires them to do only 20 slides per day per technician. So what if we receive more than 20 patients in a day? So right there is a lab technician doing diagnosis. And you see right there, he's looking through the microscope, straining his eye, looking for the pathogens. Malaria pathogens, for people who have done microscopy, and I'll show you an image of it, they are very small. So you have to really strain your eye to be able to view the pathogens. So it is very eye straining. So what is our concept detail at the AI lab to improve microscopy? As Ocular suggests to the name, we wanted to create an enhancement to the eyepiece of the microscope. So we came up with a 3D printable adapter that we attach onto the eyepiece of the microscope. This adapter is able to accommodate a smartphone. We all know that we have these days very many smartphones flopping the markets in the developing world. That means there is readily available smartphones, but there is also a big number of microscopes. So if we can be able to combine the two technologies, then we can be able to improve diagnosis. So the adapter right there is what you're seeing right here, and that is the setup that we came up with. It has got an attachment that allows almost every type of a phone to be slotted into, so long as you align the smartphone to align with the focal point of the lens, then you can be able to clearly see the pathogens through the smartphone. So long as you have a stained slide, smear slide, this time around we are using a thick blood smear slide. And the reason why we're using that is that for it provides more sensitivity when you get into detection of the pathogens than the thin blood smears. So that attachment has sliders. The sliders help you to adjust the phone to fit any kind of phone that you can slot into the microscope. It has also got some, I don't know if you can be able to see them behind here. They are the locks. So whenever you get a focal point through the smartphone, then you can be able to lock the smartphone into place. Usually when it gets to the rural areas where you may be required to do so many diagnoses, you may be required to have a charger onto the smartphone because they are limited when it comes to battery. So this setup here also allows it has a charging facility where you can be able to charge the smartphone. So right there, you see we've already slotted in the smartphone and we can be able to capture what is underneath the slide. So usually when we have a stained smear, just press it under the microscope and then you can be able to view it. So right there is the setup. So what are we trying to look at here? That is what is captured through the smartphone, and the blue label is the pathogen itself. So that is what we are looking for. Our task here is to do some image analysis. So we are basically capturing the malaria microscopic image, and that is where our analysis is going to be based on. So what we use to come up with the analysis, we have adapted deep learning to do the analysis. Deep learning is under machine learning. The reason why we use deep learning for this kind of analysis is that we don't want to get into the hassle of doing hand-engineered features. So what deep learning does is that it has a learning feature on its own where it can learn the morphology of the pathogens that we're looking for, then it can be able to return an analytics on that kind of data. So we have two implementations for this. First, we used a customized deep learning model that was based on convolutional neural networks. And then we also used a pre-trained model that was based on transfer learning. Using our custom model, for each of the pathogens that we found in a slide, we achieved around 100%. So the ROC was around 1.0 accuracy. So to throw more light on that, those were our rock accuracies. We achieved, we did for malaria, tuberculosis, and hookworm. So when we juxtaposed that to the images, the white ones are the ones that were annotated by the medical technicians themselves, and then the red ones are what the algorithm returned as true positives. So using transfer learning, we came up with a server-end application which could allow you to load an image and then it could return a detection at the end. So that was the image before detection, and that was the detection that was returned. So we achieved around 98%, though there are some parasites that were returned with a low accuracy, and we are working on how to improve that.
So why look at antimicrobial peptides? Why is this important to do? So recently, in the last couple of decades, there's been a marked slowdown in the development of new antimicrobial drugs, but a marked increase in the cases of antimicrobial resistant cases around the world. So in the US, the CDC quotes that there's over 2 million infections per year and over 23,000 deaths from resistant bacterial infections. So that's typically a case where you might go to the hospital to have some surgery, and while you're there, you get exposed to one of these, like a MRSA strain, for example, and you get sick. So this is a major issue around the world. The WHO has been putting out reports saying we need to develop more antibiotic drugs. So that's sort of the goal and driving force for this project. So what are antimicrobial peptides? So these are naturally occurring short peptides, typically somewhere around 30 to 50 amino acids in length. They're found in all phyla of life. So here's beta defensin 1, that's in human. This is meganin, which is in frog. Orelin, this is in jellyfish, and another human one called cathelicidin, LL37. So people who have defects in this peptide often will have dental problems or even skin diseases like rosacea. And so these are interesting. People want to use these as templates or jumping points to develop new types of drugs. And to date, there hasn't been a ton of success in terms of getting drugs to market off of these, but these are in phase 1, 2, and 3 trials. And so these peptides are quite diverse in their sequence. We still don't know 100% on how they work, but there seems to be sort of a common consensus that they tend to be amphipathic, and they're attracted to the negatively charged surface of a bacterial membrane. Once enough of them are there, there's a lot of different hypothesized sort of routes that they can take to kill the bacteria, but a lot of them often involve damaging the membrane of the bacteria and lysing it. So we're still trying to figure out more details in terms of how you can predict which peptide is going to do which of these modes of attack. But the project I'm talking about today specifically is just trying to predict, is this unknown peptide potentially an AMP or not? And the idea there is that it's very expensive to manufacture these AMPs and test them in the wet lab, so we want to be able to take a very large list of candidates and whittle that down to something manageable that somebody in the wet lab can actually test. So that's sort of the goal here. So there's a lot of different prior work using computers to predict or classify if a peptide's an AMP or not. Generally speaking, people have sort of used support vector machines, artificial neural networks, and sort of traditional machine learning to look in this task. They're typically sort of using things like sliding window averages of physical chemical properties, and by that I mean something like charge or hydrophobicity. But one issue that I often see again and again is a lot of people don't make their code available or their full data sets available. That's getting a little bit better, but I think that's something that the field needs to work on a lot. Currently there isn't really a gold standard benchmark data set here. That's something that's sorely missing. So, this range of accuracies, here's some example of previous work. These numbers are all on different data sets, so it's not really an apples to apples comparison here, and that's unfortunate. Typically today I'm going to be talking about Matthew's correlation coefficient. If you're not familiar with that, all you need to know is that it's a metric of how you're doing that goes from -1 to 1. The higher the number, the better. So 1 would mean that you have perfect predictions, you're getting all the true positives, all the true negatives, all that. 0 means you've got some problems. Maybe it's time to go back and retrain your model. And -1 means you probably actually flipped your labels. Maybe there's a cut and paste error. So I'm going to be talking about a model we recently published last year in bioinformatics. And we chose deep learning because it's sort of the hot topic right now. Maybe you've heard that deep learning from Google's company in the UK beat Lee Sedol. He was like Korea's #1 Go player. There's a lot of new exciting developments going on in the deep learning space. So we wanted to try that out for AMPs. So there's a whole bunch of different flavors of deep learning packages to choose from. We ended up going with Keras, which is a sort of a Python wrapper for TensorFlow, which is Google's deep learning framework. And these are all free, all open source. And this is sort of a smaller data set that I'm dealing with, and we were able to do all this just on a laptop. So just to emphasize, these sort of techniques aren't limited to people like Google. Any student with a laptop could be using these technologies for free, and the new ACE Center, that the new supercomputer being built there, could easily handle much bigger and more challenging large data sets, for sure. So just the gist of a deep neural network. Basically, in our case, we're trying to classify something. So here's just an example. If you're trying to classify an image with a number on it, so a number 0 through 9, you're going to have some sort of input. You're going to pass it through different layers in the deep learning setup that you have. And typically, it's going to be doing some sort of different processing along the way, and it's going to spit out a prediction. And this is sort of the model that we use. This is actually a model that's been very popular in sort of text mining. So one criticism of deep learning is it's sort of, some people say it's sort of a black box. Like it spits out answers, but you don't really know what it's doing, what it's focusing on, what sort of is the decision process that it's making. So we included an embedding layer, and that's used a lot in text mining. And the idea there is we just wanted to see sort of what is the algorithm doing when it considers the different types of the 20 natural amino acids? So, is it considering certain amino acids more similar or more different to each other, that kind of thing? As we'll see at the end, this embedding layer allows us to make some visualizations of how it's considering these amino acids, which is quite helpful. So all of our peptides were less than 200 in length, so we basically assigned a number to each of the natural 20 amino acids, put those into a vector, and if the sequence was less than 200, we just padded it with zeros at the front. The network quickly learns to ignore these. So we put those into the embedding layer, and then we move on to a convolutional layer. So I can explain more details on that in a break, but basically we're sort of averaging out some of those features. We move along to something called a max pooling layer, and so that would be sort of like if you had a certain window and you're sliding along it, and you're going to pick the largest number in that window and then move along. So you're actually going to shrink the number of numbers in your vector after that step. The final layer we had was called an LSTM. That's the long short-term memory. That's a type of recurrent network with a weird name. The idea with that basically is it tries to remember helpful patterns along the sequence, and it can forget sort of junk, stuff that it's not finding helpful for recognizing the pattern. And it doesn't have to be at sort of set intervals. It can recognize your pattern sort of at different lengths, and that's quite helpful and very popular in text mining. And then at the end of the day, we pass through a sigmoid function, and that just takes our prediction and stuffs it between 0 and 1, so we actually have a prediction probability. So we decided anything that's greater than 0.5 to be an AMP, and anything that is equal or less than that as a non-AMP. That's our LSTM. So where did we get the data to train on? So we used the APD3, the Antimicrobial Peptide Database 3. We used only peptides that were longer than 10 amino acids in length, and we removed ones that shared greater than 90% sequence identity to reduce some of the redundancy. Unfortunately, one thing that we also really need in this field is people don't publish their negative results. So people love to publish when they find a new AMP. They don't like to publish, oh, this peptide actually didn't kill bacteria. That would be great to have, but until we get that, unfortunately, right now the best we can do is taking, it's sort of standard in the field, to take peptides from Uniprot or a similar database, use some keyword filters to remove antimicrobial and that kind of thing, and then filter those. So I'm considering my non-AMPs as ones taken from Uniprot that don't have certain keywords in it. And then we're going to randomly split these up into a training, tuning, and testing set. So I had the same number of AMPs and non-AMPs for all of these. So 712 for training, 354 for tuning, and 712 for testing. And so how did our model do if we were to compare against other publicly available methods? So if we took our testing data set, put them into their server and look at the results and compare it to how we did at the end of the day, we get better results on all the different, almost all the different metrics we considered. So to orient you here, our first column here is the different methods. Our methods at the bottom. The website for these that you can check out on your own is listed here. So we have sensitivity, specificity, accuracy, that Matthew's correlation coefficient I mentioned before, and the area under the ROC curve, like we saw in the previous presentation. So in our case, sensitivity was highest for this CAMP server. They used random forest. But for all the other metrics, we beat the competition there as marked in bold. So particularly in the MCC category, we're doing quite a bit better than the competition. So here's the ROC curve. For the black line here, which is the top left here, which just means that we're doing the best. So I mentioned that embedding layer. So sort of, can we get into the mind of our deep network a little bit? And so basically we started, with a 128-dimensional space. We can't visualize that as a picture, right? So we use something called T-SNE, which is going to take that high 128-dimensional space and project it down into 2 dimensions, something that we can actually put into a graph, which is very helpful. And so these relationships, basically, the closer the letters are, these are amino acids here, the closer the amino acids are to each other, the more similar the model considered them. The further away they are, like for example the cysteine here, the more sort of different it considers it. And so it learned this relationship on its own, and it's kind of interesting here. There's a lot of sort of known physical chemical properties here that it just sort of figured out on its own. So for example, we have some negatively charged peptides here, or amino acids here. We have sort of antipathic amino acids over here. We've got some sort of, I call the oddball amino acids that are just a little bit different. Over here, for example, tryptophan has a very bulky side chain. And cysteine, which is involved with cysteine-cysteine bonds. So I thought it was very interesting that sort of the model on its own figured out some of these well-known features that you wouldn't find in any chemistry textbook. So you can check out our model. We have a free-to-use AMP scanner website, ampscanner.com. Our data sets are available to download. I have contact information if you want the code. It's free, open source. I'll send it out to you. So check it out. I try, this is sort of a side project here, so I tried to sort of update it once a year at least. So I recently updated it in February when new AMPs become available. It's fresh up. It's been freshly renewed right now. For the sake of time, I can't go into too much detail here, but I just finished sort of a pilot study with some other colleagues, a new grad student in Dr. Marta Shehu's lab, who was where I did my postdoc or my graduate work. And so we were kind of interested in creating a generative model. Can we reuse this new deep learning network in sort of a new way? So sort of a hot topic right now in self-driving cars is making a simulated environment that the car can drive through. So there are strict requirements for, like, in the US, our Transportation Administration says that they have to pass before it's legally going to be allowed to let these cars, drive themselves on the road, right? You have to prove that it's safe. So they're using simulated environments. So basically in this paper, we found a way to create AMP-like sequences and use our model as a judge to see if they're AMP-like or not. So we're hoping we can use this down the road to create sort of larger data sets because there's only a limited number of AMPs available. Okay, so just some next steps. We're interested maybe now that we can generate sort of AMP-like sequences to maybe try something called adversarial learning. So Google does this a lot to teach games how to play games against each other. So they'll have like one model play, say, chess against another model, and they learn from scratch how to get much better at playing chess against each other. So maybe we could have two models compete against each other to see who can be a better AMP predictor against each other, and they might get better performance overall. Also, in this case, it's great that we can say that a peptide is maybe AMP or not. At the end of the day, you still need to confirm that in the lab, the wet lab, of course. But it would be very helpful for clinicians to be able to say, not only is this an AMP, but that's an AMP that's going to work against a particular strain of bacteria that we're seeing as a problem in the hospital. So the sort of next step here is trying to predict how active this peptide might be against, say, for example, MRSA, that kind of thing. So I think that's sort of the next step in this field.
I'm called Joyce Nabende. I'm from the AI and Data Science Research Lab from Makerere University. On the formal side of the AI lab, we are trying to do research. Rose has given us one aspect, but there are several aspects of things that we are working on. We try to do these things for the end user, so we do for the people in the health domain, we do for the farmers, the smallholder farmers out there. That's outreach. We also focus on capacity building in terms of student training and collaboration. So my talk is really going to talk about application of artificial intelligence in health. I'm trying to go through the slides because there are quite a lot. And it's because now we have lots of data coming out. And if we're looking at AI machine learning, we need data. And this is being given to us now. So what can we do with the data, and especially in the healthcare domain? So in healthcare, we all know we have a lot of high repetitive work. You come, you have to go through the same procedure. I usually take my son when he falls sick, and it will be the same thing. Take the temperature, take the weight, then you can go to the doctor, and it will always be you have an infection. So sometimes I feel like I can also be a doctor anyway. The only thing is I don't know the medicine to give, but I know what the procedure is. So with AI, we want to empower the doctors to help them deliver faster and more accurate results. We want to augment them, their expertise, and assist them. We want to manage waiting times and automatic scheduling. So if we're looking at the application of AI, we'll have two main branches. The factual branch will mainly focus on the application, the machine learning techniques, either they are supervised, unsupervised, or reinforcement learning. But what are we trying to do here? We want to make sense out of the data. If we look at the physical branch, these are the physical objects that a human can have, and we've had several of these, and I'm no expert in this area, but there are several things that could do to be able to take sensors, to monitor people, robots for delivery of surgery. So there are lots of things in the physical branch of AI in medicine. So I'm going to try and talk through some practical applications that we can have, especially in the developing country, and then I'll finish up by discussing some open challenges of what can we do based on these very good things that are existing out there. So we have electronic medical records. So of course right now we are trying to embrace this, and many of our hospitals are embracing the use, for example, of technologies like OpenMRS and the DHIS2 for collecting, validating, analyzing, aggregate data, and patient-based statistical data. So in terms of AI, what can we do? We can look at building AI applications on top of this data for complex predictive models. We have a student in the lab who's trying to look at how can we analyze the DHIS2 data to kind of build predictive models for pneumonia in children. So there's that rich information that's there. Can we build AI applications and algorithms on top of that to analyze this kind of data? Cloud computing is a rich aspect that is also growing and being embraced right now in our country. If we have these health medical records, can they be maintained in the cloud? How can we be able to leverage on machine learning, connected devices, IoT, to be able to provide ways of capitalizing on these different resources that are existing? So instead of having the data here and it's remotely accessible, how can we be able to use machine learning? And there's, for example, a project that's using interactive voice response calls for disease management. So you just call in and you're able to be able to get, for example, a diagnosis. And this is something that we could especially use in countries that are in our country, for example, for regions that are hard to reach. Can someone be able to leverage on the connectivity that we have in those regions? For example, for interactive voice responses between the physicians and the patients. Mobile health, we've had very many applications in Uganda being built around mobile phones. They'll be used for all sorts of things to improve provisions of health services, to communicate to patients through the SMSs. And so for us as a lab, one aspect that we always capitalize on is the aspect of image recognition, and we'll talk about it later, and computer vision. Can we be able to take a phone, use it to take an image or a photo, and then can we be able to do a disease diagnosis? One aspect which I found interesting as I was reading is something called
pattern recognition, where AI applications on the phone are actually being used to listen to the sound of coughing and breathing to be able to accurately diagnose pneumonia or asthma. So you don't really need to have a doctor there. You can be able to tell based on the way the person is speaking, we had that over break, speaking or even breathing, and we can be able to have this penetration of mobile phones and then build applications on these mobile phones to be able to help in diagnosis, in disease management, for example. The other area is expert systems. In resource-poor context, this can be able to be used to support health programs in very many ways. They can help in choosing treatment plans, as it's being done in the high-income countries. But if also a human expert is not readily available, can we build expert systems, for example, through chat boxes or virtual assistants to kind of create and leverage this gap between patients and clinicians to improve patient experience and also reduce physician overload. So MA, artificial intelligence applications, can be built around this area to be able to reduce the physician's workload. Disease spread, we've had students also in our lab looking at the spread of diseases across Uganda, and here they can leverage on the availability of the data that we have. For example, the electronic health record data, telecom data, demographic data, and trying to put all this together to build Bayesian models that can be able to model disease incidence and surveillance. So AI can also be applied in that area for disease spread. NLP, natural language processing, is also a very good application area. We could look at the clinician's notes. If we have these notes written down, can we be able to extract information out of that? Looking at are they following the clinical guidelines that are provided? For example, the lab reports that are being seen. Can we be able to develop human-to-machine language instructions that clinicians can be able to follow? But also, one of the interesting things right now is that we are now moving towards, or many people are now embracing social media, Twitter, Facebook, and so you can use NLP there to be able to search for surveillance and detect surveillance or disease outbreaks based on tweets, based on what people are putting out there on their social media handles. So that's also, I think, a very good area that we could capitalize on in terms of AI application. Signal processing, I think I talked about this in terms of the physical aspects of AI. We have mobile phones. We have the smart wearable devices that will collect all kinds of digital data. So in terms of AI, in the resource-poor settings, this is really promising because it can be paired with machine learning principles and the cloud computing that I mentioned earlier to be able to improve in patient care and patient diagnosis. Computer vision, this is very key to us as a lab. Can we be able to use computer vision and image processing to provide an easy solution for medical diagnostic imaging? Maybe some challenges, especially for us in a developing country. If we're looking at machine learning, we always rely on data. So how can we be able to get high quality data sets that we can use to train algorithms? Many of them are not easy to get for many diseases and conditions. Such data sets are hard and time consuming, and I guess this also relates to the bits that I'm going to move quickly to of privacy. Yes, privacy as well. People are like, no, where are you taking our data? What are you going to do with our data? So people want to maintain secure, transparent health records. How can they be able to do this? So that's a very big challenge, the privacy, but also the data set that is in the format that you'd like to push it into your algorithm to do what you want to do with it. Recently, the president signed a law, the data protection law. It's not a bill anymore, so that highlights different things that you could do with digital data. And so I think it would be good that if we are moving into that space that we can easily comply with these government healthcare standards. Just to go back one more slide, NLP. If we are trying to look at NLP, it really relies on corpora, language corpora. But most of the settings that we have in our country are usually handwritten, and they're even written in the local languages. So if you went to deep down there, you might find that things are not in English. So you kind of need a lot of translation between English and maybe Luganda to be able to actually use this kind of data set. So the compiling of this corpora can be a big challenge if you want to apply machine learning. Mobile health, internet connectivity, yes, we are improving. We have 2G, but 4G, 5G now, but you might go to some places that, suddenly just switches from 4G back to 2G. And so it might be a little difficult if you're trying to use your phone to capture images or to capture data and send it over to the DHIS2. How do you do that with slow connectivity? So that could be one of the challenges that has to be addressed. Then, importantly, patient-doctor comfort. If you're saying that you're now going to have us using robots, are patients very comfortable with machines, with applications, and sometimes also, as they mentioned in the first panel, doctors might feel threatened. Are we going to lose our jobs? So that's something that we kind of have to train people to use, and I'm not saying that when we have AI systems, it's taking away their jobs, but it's just aiding and their work. But also from the patient perspective, I think that would be, for example, a challenge in terms of their privacy and their comfort.
Hello, everyone. It's my pleasure to be here and talk about a topic that I have a strong passion for, and it has to do with microbiome, the human microbiome. And I have to say, my role at the institution that I work at NIH is really on data analysis, and I work with multiple collaborators, but today I decided that I'm going to take a higher view. I'm going to basically go through what we knew about microbiome a few years ago and move forward slowly. So it's going to be more of a tour. So microbiome. When I started graduate school in 2000, I never heard of this topic, never heard of the term, and I studied molecular microbiology. So through my 5 years in my PhD program, never heard the topic microbiome. So it is pretty new, although it was described many years ago, but it wasn't until about 2001 that Joshua Lerberg defined it, and it's been now reported in many publications that he was the person who coined this term, and although that may not be true, but anyway, so the definition that he posed is that it is the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. But looking back, if we go to the 1600s, we have that, we probably all remember from our biology, that Anthony van Leeuwenhoek, who developed a microscope, he was observing from his own samples in his mouth that there were animalcules that were in such enormous numbers that all the water seemed to be alive. That's how he described it. And he made many pictures, drew a lot, and we have books that describe those pictures. But it wasn't until about 200 years later, when then we have Pasteur and Koch, that were describing more of how those microbes affect human health. And they were identifying how pathogens were involved in anthrax and tuberculosis and cholera. Later on, by 1910, we had a different view. This French investigator, he described and theorized that health could be enhanced by manipulating the microbiome. He didn't call it microbiome at the time, but he did express how yogurt could be a good treatment. And that's very interesting because, I don't know about you, but everything that I learned when I was little is that germs are bad. And it was that you had to wash your hands a lot, right? And so, but it was described early on how microbes could be beneficial. Later on, there were many other investigators working on other diseases that were identifying pathogens. And then, again in 1950, another investigator, René Ducros, he was very passionate and a very deep thinker, almost a philosopher. So he he sent many comments about health. But one great contribution is that he developed a germ-free animal model. He started working with mice and were able to infect or transfer microbiota from one mice to another and see how it was actually affecting the health of the other mice that received the microbiota. Again, that happened very early on in the 19th or mid-century, but it wasn't really popular until recently. Now, in about 1980s, then we developed or started using more of the RNA sequencing technology, the ribosome RNA, which is a gene that is very conserved in bacteria, and we're able to use it to classify bacteria, and that had a strong impact as well as other technology. But I wanted to also highlight another example that I find super interesting that I learned actually early in grad school when working on H. pylori. And Helicobacter pylori is in the gut of many of us in here. It should be, hopefully, unless probably the children in the US, maybe not so quite. But those of us that were playing with dirt early on in life, most likely have it, and we don't have any problems with it. But it turns out that people thought that gastritis and ulcers were really the cause of stress, and the stomach was so acidic that the bacteria wouldn't survive in it. And it wasn't until Barry Marshall and Robin Warren decided to take a bold approach and swallow the culture of H. pylori and prove that he actually could get infected with it, and then of course also proved that with antibiotics he could get cured, that then he actually made a contribution of how, again, microbes contribute to disease. But interestingly, around the same time, Dr. Martin Blaser from NYU, who's had been studying H. pylori as well for a while, kept also insisting about the antibiotics until suddenly he started doing more and more research and realized that in fact, yes, H. pylori may have a detrimental effect in some adults, but in some children it may cause more harm. It may cause more harm if we eradicate it, if we give antibiotics early on in life and the children don't have H. pylori. So now, in recent years, it's been fascinating because yes, H. pylori has been implicated with cancer even, but actually now there is the theory of the hygiene hypothesis, contrary to the germ theory early on, where it's thought now that early exposure to germs help a child's immune system develop resistance. And that's what we're learning, and the investigators that have been working on at NIH have been looking very closely at the immune system and the role of the microbiota. And we now appreciate that the human microbiome is very much essential in many functions in our cells, and that there's a very good symbiotic relationship, and that of course there is an influence by diet, exercise, medications, and so on in our communities, and that's very important. I want to just quickly highlight that there's so many bacteria, microbes, viruses, protozoa in our bodies, and not only that there's so many more microbial genes than what are human genes. Apparently, there are about 20,000 human genes, but in reality, there could be up to 20 million microbial genes. So you can imagine that there's a lot being produced in our bodies that will have an important effect. Therefore, we want to definitely pay attention to what the microbiome is doing. So it was during the years of 2007 that the NIH funded this project, the Human Microbiome Project, with the idea of exploring in more detail the relationships of microbiota with human health. They looked at many genomes, used technologies of sequencing to explore which taxa were there, and determine the relationship in between disease and dysbiosis, which really is an alteration of the communities. They developed tools as well and explore about legal issues. But they didn't really look at in this stage much into disease, which is why then they went on and worked on the second phase, IHMP, or integrated HMP. But yes, HMP1 did produce significant findings, and one of them, for example, is that there's an increased risk of cardiovascular disease due to some metabolic byproducts. As an example, there's many others. But also that even though there's differences in between subjects in their composition of their microbiota, there could be many similarities in their metabolic pathways. So this led to more and more research. There's labs across the world that are studying multiple microbiomes across the body and associating the changes that they see, or trying to understand what is this cause or effect of the changes in the microbiota with many diseases, that even obesity. And now I would like to highlight some common methods for studying the human microbiome. As I mentioned earlier, the model organisms can be used, and that's this germ-free technology that I mentioned that was used back in the 50s is now being used heavily. There's bacteria cultures, which is a challenge because not every bacteria can be cultured. Now there are many new developments or better methods to culture bacteria recently. There's in vitro studies that can be done, and culture-independent methods. And here is where I'm most involved, where there's sequencing of the bacteria that maybe cannot even be cultured, and we can actually compare the communities of the various organisms. And to highlight the first one, the germ-free models, this is super interesting because you're trying to really explore what happens when you have a very well-defined microbiota, where it's either free of all microbes or defined, or free of specific pathogens. And then you can introduce some others and see what happens in the mice at the level of the immune system or health in general, diseases, weight, etc. There's a few examples of those. For example, this paper back in 2014, in this case, what they did is that they took a mice and they gave antibiotic. The antibiotic caused an increase in weight. Then they took the feces and transferred them to a germ-free mice, and they were then able to carry on and see that the mice, just by taking on the microbiota from the one that gained weight, also showed the effect of gaining weight. So it led, early on in life, it led to perturbation that could have that long-lasting effect, and that's very important that we're understanding that now. There's another more recent publication, this is from last December, I believe, and where they looked at obese mothers and how that increased inflammation and susceptibility to NAFLD, which is the fatty liver disease. And what they did is that they took the babies from obese or lean mothers, and I'm showing you the picture of the obese, and then they took the microbiota, transferred it to the mice, and were able to see that that caused an immune effect in a way that was consistent with the phenotype of the non-alcoholic fatty liver disease, and also that if they were being exposed to the Western style diet, they were gaining weight. Another method that I want to talk today then is the omics method. And this is where we have very a good availability of data for metagenomics, and we can then, or what we have been doing is that we have been collecting a survey of where in each of the microbiomes or the different tissues we have certain classes of bacteria, and that was really the early years of the microbiome studies. Now we want to really go beyond that, just not characterizing only, but I should say, this is a free advertisement to a free tool, is that we developed this tool in IID in our group, and it's called Nefeli, and if anyone would like to use it, it's totally free, and it's for data analysis, for metagenomic analysis. But then we also have other omics, like metabolomics, and it is super interesting because you're now assessing the metabolic profile of everything that is in the sample, the bacteria, in looking at what they're being produced and how that is influencing, of course, the immune system. So the immune system is scanning constantly, and things like butyrate, for example, leads to an anti-inflammatory response, which is awesome, but therefore we want to increase the bacteria that are producing the butyrate, and it is believed that eating fiber, eat your beans, and maybe then you'll have that good bacteria producing butyrate and can reduce inflammation. Metaproteomics. So if you have a sample that you can extract all the protein from the host as well as the bacteria, you can also determine not just who is there, but what are they being produced, what are they producing. This is done like through the metabolomics using mass spec technology. Metatranscriptomics is looking at the gene expression of, again, the host or the bacteria. And there's examples of this recently. There's a paper from the end of last year where they were looking at oral squamous cell carcinoma, and they looked at transcriptomic analysis and identified that in those communities or in those samples with the OSCC, there were genes that were being expressed that had to do with virulence factors in bacteria at those tumor sites. This is a way of incorporating the data from the expression of genes, not just who's there. Machine learning is another tool that we've been using, or that some groups are using, of course, and I'm glad that everyone has mentioned it. And basically in here, as an example, they were looking at IBD diagnosis and a response to a particular drug, and they used the random forest classification in this case to really predict the accuracy of the Crohn's disease or colitis. So another example of how that can be used. But it shouldn't be the end of it all. We should actually move to more, which is integrated genomics. There's a lot of data that is becoming available, and there's a lot of efforts to create projects that integrate genomics, almost done. And what's next? We want to manipulate the microbiota so that we can, like they were trying to do back in the days, using yogurt, or now these days, we really have many methods, like targeting, antibiotics, of course, which we want to make sure we are reducing as much, and then probiotics, supplements, and transplantation, like fecal matter transplant.
I was invited to give an overview of activities happening at Untitled that are utilizing omics approaches, but I will try to stay in my comfort zone of virology, especially HIV research. So we know that prevention is actually working for HIV. We know what what is appropriate. But unfortunately, we are continuing to see a lot of new infections, despite the fact that we know what is effective. And of course, treatment has been rolled out as prevention. But at the moment, there's a lot of emphasis on trying to understand our local epidemics geared towards prevention. The other problem at the moment is the emergence of drug resistance. It is a very huge problem at the moment. So at the moment we are utilizing phylogenetics to really understand our epidemic for prevention. So how has phylogenetics been made possible at the moment? So as you know, at the moment technologies have tremendously improved, and we have a lot of sequence data which is available, and a lot of socio-epidemiological data which is available. Sequencing technologies have tremendously improved. As you may see, at the moment, next generation sequencing is generating a lot of data, and this has enabled us to really understand transmission networks. And of course, the availability of high performance computing is a plus. So for us, who are utilizing phylogenetics and bioinformatics, we are ideally interested in identifying these transmission networks, especially people who are transmitting the virus within a population, but also we need to know who are those that are infected, and that is very appropriate for prevention. So at the moment, we are working with the ministry. We are doing a lot of surveillance of drug resistance in the population. So we are working with central public health laboratories where the viral loads among people on treatment are being done. And what we do is we do surveillance of drug resistance among those individuals who happen not to be suppressing the virus. So we have really generated quite a lot of data, sequence data from these patients, and we are currently utilizing this data. So at UVRI, in this building, which is funded by the Wellcome Trust, we do have an HPC facility where we are storing most of the sequence data that we are generating, and we link this data, the sequence data, to the epidemiological data for these patients. And that has enabled us to really try to look at the transmission networks within the different populations. So I will talk about a few surveillance activities and studies that we are doing. We're doing quite a lot, but I would simply talk about what we're doing in the fishing communities. We know that these are very high-risk individuals in the fishing communities. So these are some of the populations that we are looking at. Those are landing sites, a typical landing site on the shores of Lake Victoria. Those are the fishing activities. But we also have activities in Kampala, working in the female sex workers in Kampala. But for this presentation, I would simply talk about the fishing communities. So these are the sites where we're working. That's the map of Uganda. This is Lake Victoria. So we are working along these landing sites around Lake Victoria, but also we are working in the island populations to look at transmission networks in these populations. I will not talk much about the methodologies. This is where we have our sequencing core, the labs, the activities that we do, the sequencing as well as the phylogenetic analysis, as well as looking at the phylogeographic analysis of these populations. So, in the fishing communities, first of all, we all know that it's a common saying that the fishing communities are driving the epidemic. But the question is, we don't have hard evidence to show whether these people are actually driving the epidemic. Now, so we were thinking that these fishing communities were reservoirs of HIV transmissions. So we thought that these fishing communities are the ones who are spreading the virus. So we wanted to identify the transmission networks in these fishing communities and also in the general population to see whether there are linkages between these communities. So what did we find? So when we did phylogeographic analysis, to our surprise, we found that the transmissions are actually coming from the general population into the fishing communities and not the other way around. So you can see the viral migrations into these fishing communities. Now, so the question is, and we have shared this information with the Ministry of Health. This has implications in as far as public health is concerned. Where do you target your interventions? Do you simply get into the fishing communities? But at the moment, we're seeing that actually it is from the general population, from the mainland into the fishing communities. So again, there's need to refocus where you need to intervene. So using HPCs and the databases, we have now embarked on efforts to try and institute some kind of near real-time monitoring of the epidemic. So as we get sequence data, all the sequence data that comes in, it is put into the database, and then we can also have a real-time kind of monitoring to see in case we are seeing new infections. For example, if we do have an individual who is newly infected, and this individual has drug resistance, assuming this is a new case, okay, and this person has drug resistance. So you're able to identify these transmission networks, and then you're able to move into these populations for proper epidemic management. And these are initiatives that we are currently working on, again with colleagues from Makerere. I'm happy to see you, Daudi. And so we are also part of a big network, Pangea Consortium. This is phylogenetics for networks for generalized HIV epidemics. This is a Bill and Melinda Gates Foundation funded initiative. It is a consortium of several research groups in Africa. In Uganda, there's RACAI, the MRC, UVRI. We have colleagues from Zambia as well as Botswana and South Africa. So the initiative is targeted to come up with over 20,000 full HIV genomes within sub-Saharan Africa. And this is intended to really understand the epidemic in sub-Saharan Africa, but also look at where they can target interventions. So we have contributed to this initiative, and one of the key findings we have seen from Uganda, we've generated over 1,000 near full genomes from HIV. But we also included about 100 samples from the early HIV epidemics. These were samples which were collected in the early 80s. And so we've been able to do subtyping. And what have we seen? At the moment, our HIV epidemic, it is predominantly subtype A and D. And those are the ratios. But what is very interesting is that we are seeing that currently over 50%, these are recombinant viruses, mainly between subtype A and B. And this is showing that, in fact, individuals get multiple infections with different subtypes. And this has a lot of implications for vaccine design and development. I will not get into the vaccine work that we're doing. However, what we noticed, the samples that were collected in the early 80s, very few were recombinants, and we've seen a significant increase in recombinant viruses. That again has very serious implications for vaccine design and development. So, I will briefly talk about some of the other omics activities at UVRI. So there's a department working in emerging and re-emerging infections, the Adversarial department. It's doing quite a lot of work in this area where they're doing a lot of surveillance activities for these emerging infections. We are working with colleagues from CVR, that is the Vaccine Research Center in Glasgow. This is Emma Thompson. And from the metagenomics analysis that has been done, among patients with acute febrile illness, we have seen a lot of co-infections among these individuals. We've seen individuals with co-infections with HIV, hepatitis A. We've recently reported Ledante virus and a new virus called Adumi virus, and a lot of other suffered viruses. And then this is the report, the recent report of the Ledante virus, which was reported recently. The first case was in Dakar, Senegal in 65. The other case was in 77 in Wales, and the most recent in Uganda was again identified. So what are the challenges that we're facing at the moment? There are a lot of challenges with, of course, phylogenetics. As you know, you need a lot of huge sampling in your population to really have meaningful interpretation among these infections. Lastly, you still need high computing space as well as data storage, because a lot of data is coming out. But also, there are a lot of ethical challenges. When you talk of HIV transmission networks, one fear comes out, intentional transmissions of HIV, and we've had some legal challenges at UVRI concerning this kind of transmissions. And I would like to acknowledge a lot of funders and collaborators. Thank you so much.
Thank you. As we wait for the slides, I want to briefly introduce myself. I come from the Infectious Diseases Institute in Makerere, and I see many of my colleagues in here. I head the statistics unit, and today I just wanted to find out, I just wanted to let you know how we are applying data science in the field of HIV clinical research at the IDI. Okay, I'll skip most of these. As a statistician, I just wanted to brag that statistics was one of the oldest fields as compared to data science, but it's a mix of so many fields that kind of combine data science and how it's applied in the current field. So the IDI is a center of excellence that provides care to over 300,000 patients. And currently in the IDI Mulago alone, we have over 9,000 patients who are actively receiving care. We also have a 10-year cohort of patients who are being followed up with structured laboratory and clinical monitoring. And IDI also serves as an implementing partner for the CDC in the country. And in that realm, it provides care and treatment to over 4 regions in the country. And this provides massive amount of data that we collect among these patients, repeated labor data and labs, and so much information about opportunistic infections among these patients who are receiving care through the IDI support. And we also have access to EMR database through collaborations with the MOH and institutions like the Monitoring and Evaluation Departments at the School of Public Health. And through this data, we can have access to dashboards and information that is collected among patients, and that's available to the nation on the web. So in order to harness this large data and use this to develop models using data science methods or machine learning, we need to have the data, which we have. We need to have brains in addition to the computer to develop high models that are really functional. And what has also been previously mentioned with the prior speakers is that the outcome of your modeling is how good your model is. And these have been showcased before with different performance indicators like the accuracy, precision, and sensitivity and specificity of your models. So one of the ways that we have attempted to use data science methods or machine learning is to try and predict mortality after ART initiation. So ART is antiretroviral therapy that is given to patients who are HIV positive. And we know that over the years, mortality has decreased because of the rollout of ART, but we still see a good number of patients who die right after mortality. Not that death is immortal, we all have to die, but just predicting mortality after ART remains of great importance because you find out how you can prevent this as clinicians. So our aim was to use machine learning methods to predict mortality among patients after 5-year enrollment in an RCT. And the data comes from a small portion of patients. You might wonder why we only use this given the large resource that we had. But like we say, data is there, but we need to look at the quality of data that comes out. So we split this data. This data comes from about 400 patients, and we split it into 9/10s for training and use random forest and support vector machine models to kind of predict who dies right after ART initiation in a 5-year follow-up. And when we did this using the two models with the support vector machine and the random forest, we see that these models could accurately predict who is going to die and when who dies right after ART initiation. And these findings have greater implications on how you follow up patients right at the time of ART initiation. And the support vector machine performed relatively well with an accuracy of 93 and an error curve of 97. But the random forest model performed even better with an AUC over 99. Another way that we have attempted to use machine learning to predict in our settings, again, is to try and predict who is retained in PrEP programs in our settings. PrEP is generally giving antiretroviral therapy to HIV negative people to keep them negative. But one of the things we see is that persons start PrEP, but at the time of following PrEP, at the time of initiation of PrEP, you see a lot of disengagement along the way. Currently in Uganda, PrEP is given to people who are known as high risk and priority populations, probably some of the populations that are seen by DEO in the islands. But along the way, they drop out of the PrEP cascade. You start with about 100%, but after 12 months of follow-up, you only have 22% of your people currently still on PrEP. So we wanted to predict who drops out of PrEP because, again, if you have this information, then at the start, at PrEP initiation, you can roll out who's likely. You can identify who's likely to drop out and then maybe focus more interventions on this subpopulation. Again, we used machine learning methods. We employed the XGBoost algorithm using Python to predict retention following PrEP. The data was obtained from the PrEP dashboard, which is available. And at the time we extracted the data, we had close to 8,000 patients. And again, we split the data into a testing and test data sets. And we evaluated the model again using the same performance measures. And using that measure, we were again able to predict who is likely to drop out of PrEP at the time they initiate PrEP. And the model performed fairly well with an AUC of 98%. And we also noted that female sex workers and persons less 18 to 24 years were likely to drop out of PrEP programs. And the next step in all of this work is to design a risk score that can be used by health workers to identify who is going to drop out of PrEP. And other ongoing projects that we are working on as a stats unit at the IDI is to develop a risk score for predicting disengagement from PMTCT programs. This is similar to what we see in PrEP. Mothers start ART, but they drop out quite early because of several reasons. If we can identify this population right from the start, then we can design interventions for keeping them in care, and similar to tracing in the community. So our conclusions is that data science methods can be applied in HIV clinical research. They come with a lot of caution because, like the previous speakers have mentioned, garbage in is garbage out. The quality of data really, really matters. Most of our data is coming from the Open MRS and the ICEA, which is an integrated clinical platform that's developed in IDI that has considerably better quality data. But all of these really depend on what you put into the model. And our results show that if we work together with clinicians and the AI group and all other groups that have been mentioned here, we can definitely come up with solutions to existing problems in HIV clinical research.